Today we'll see a histology of thyroid gland. The thyroid gland, which is situated in the anterior region of the neck, consists of two lateral lobes connected by a narrow isthmus. The isthmus lies over the second to the fourth tracheal cartilage, and lateral lobes lies in relations to the superior part of the trachea and to the inferior part of the larynx. Frequently, a median pyramidal lobe, which extends upward anterior to the larynx, is present in addition. The gland is developed externally by connective tissue capsules which is continuous with the deep cervical fossa. Under this, there is an inner two capsules which is thinner and which adheres closely to the gland. Delicate continuous of inner capsule extend as septa into the gland and divide it into indefinite lobes and lobules. Follicles, the structural unit of a gland, composed lobules. They vary greatly in size depending upon the degree of distensions by secretions. They also vary in shape but usually are irregularly spheroid. The follicles are embedded within a delicate meshwork of reticular fiber which also supports a closed net of fenestrated capillaries. A follicle consists of a layer of simple epithelium enclosing a cavity which is usually filled with a stiff jelly that is called colloid. The shape of the component cells varies but commonly is cuboidal. The cells are low when the gland is hypoactive, high when the gland is hyperactive, and cell height in any one follicle is uniform and the arrangement is regular. The base of the cell rests upon a delicate basal lamella. The large vesicular nuclei lie centrally or toward the base. The cytoplasm is finely granular and basophils and contains numerous mitochondria, abundant granular endoplasmic reticulums and lysosomes. The Golgi apparatus centrioles are located above the nucleus. Lipid droplets and other inclusions, particularly colloid droplets, are found in the cytoplasm of some cells. Functional complexes are features of the interface between cells and the three border is provided with small microvilli visible only with the electron microscope. Some cells possess true cilia. Colloid filling the follicular lumens, that is the fresh colloid is homogeneous, clear and viscous. However, it undergoes shrinkage during the procedures employed in the preparations of microscopic sections and may show irregularities. Space opens are present between the colloid and the epithelium, and vacuoles may occur within the colloid. The irregularities are indications of the state of the colloid and are most common in activated glands. Colloid stains basophils in active follicles, whereas it stains weakly basophil or acidophils in inactive follicles. The colloid, which represent reserves of secretions rich in a nucleoproteins, hence it is a basophilics and contains thyroglobulin and enzymes. Thyroglobulin is a glycoprotein containing several iodinated amino acids, the proportion of which vary from follicles to follicles. Thyroglobulin stains deeply with the PAS reactions. The secretory process is complex and difficult to follow. It involves synthesis of the thyroid hormone, temporary storage and release into the perifollicular capillaries. Both synthesis and release may occur at the same time. Thyroid cells removes iodine rapidly from the blood streams and concentrate it. The major part of the iodine is incorporated in follicles in an organic form. The incorporation of iodine in follicular cells and in colloid can be followed by use of radioactive iodine. The iodine within the colloid is present in the form of a diiodothyronine and triiodothyronine. and tetraiodothyronine bound to a globulin. These are component of thyroglobulin. The release of the active component also is difficult to follow. It is thought that thyroglobulin first is recovered from the colloid by a process of pinocytosis at the apical surface of follicular cells. Then hydrolytic enzymes from lysosomes then break down thyroglobulin into smaller molecules, principally thyroxine and triiodothyroxines, which pass through the bases of follicular cells into the underlying capillary plexus. The thyroid contains, in addition to the principal cells of follicle, a small populations of parafollicular cells, that is, uh, some see or clear or light cells. These cells, which lie adjacent to the follicles but within the basal lamella, do not about on the follicular cavity. Generally, they are larger than follicular cells and their nuclei are placed eccentrically. They are characterized by the presence of numerous membrane-bound dense granular throughout the cytoplasms. Immunofluorescence studies have shown that these cells are the site of production of thyrocalcitonin, or it is called also as a calcitonin. Now, in continuation with this thyroid gland, we'll also today we'll see this parathyroid gland, which is a part of a major thyroid gland, and the location is also on the same organ in the neck over the thyroid glands. The parathyroid glands named because of their association with the thyroid glands and are usually found embedded in a connective tissues capsules on the posterior surface of the thyroid. In human, usually two pairs of small yellowish-brown oval structures are present. They typically measures about 6 mm in length, 3 to 4 mm in width, and 1 to 2 mm in thickness. They are identified by their locations as superior and inferior parathyroid glands. In some individuals, that is, estimate on the frequency vary from 2 to 10%. Additional glands are associated with the thymus. The structure of parathyroid gland. Here, each glandular mass is surrounded by thin connective tissues capsules that forms with the septa extend into the gland, dividing it into poorly defined lobules and separating the densely packed parenchymal cells into astenosine cords. The extent of the connective tissue becomes more obvious in the adult as the number of fat cells increases with age. Their number increases dramatically at puberty and continues to increase throughout the life. In the elderly adult, 60 to 70% of glandular mass may be occupied by the fat cells. The septa provides a route of access for the blood and lymphatic vessels and nerves. Two basic cell types are identified in a glandular epithelium: a principal or chief cells, oxophilic cells. Some investigators identified a third transitional cell type that is intermediate in structure between the two. However, the feature associated with these cells may result from poor fixations of the tissue. The principal cells are the most common parenchymal cells in the parathyroid. They are small, polygonal cells, measuring about 7 to 10 micron in diameter. They have a centrally located vesicular nucleus and a pale staining, slightly acidophilic cytoplasm. Lipofuscin granules, large accumulations of glycogen, lipid droplets, and small, dense membrane-limited granules, presumably containing the storage form of a parathyroid hormone, are seen in addition to the typical cytoplasmic organelles. The oxophilic cells constitute a minor portions of the parenchymal cells. They are distinctly larger than the principal cells and have intensely eosinophilic cytoplasm. Their nuclei are often smaller and more heterochromatin than those of the principal cells. Ultrastructural studies have revealed that the eosinophilic staining of the cytoplasm is due to the presence of numerous large mitochondria. Morphologically, the cells are further characterized by limited amount of endoplasmic reticulum, a small Golgi complex, and the absence of secretory granules. Now we'll see functions of this thyroid gland and parathyroid gland. First, we'll see the functions of thyroid glands. The most striking effect of thyroid secretions is its regulation of metabolic rate. Thyroxine increases cell metabolism and thus it is concerns with the development, differentiations and growth. Hypothyroidism in the infant leads to cretinism. Hypofunctions in the adult causes max edema. Symptoms in both conditions are due to the reductions in a metabolic rate and may be removed by the administrations of thyroid. Hyperthyroidism leads to overactivity and sometimes is complicated by the development of exophthalmic goiter. In hyperthyroidism, some follicles become enlarged and follicular cells increase in height. Surgical removal of a part of a thyroid or the administrations of antithyroid drugs or radioiodine reduce the metabolic rate. The thyroid gland elaborates in addition to the thyroid hormones, thyrocalcitonin, a product of a parafollicular cells. This hormone is polypeptide that actively lowers the concentrations of calcium in the plasma by the direct action on bone, inhibiting bone reabsorptions. Hypercalcemia is the stimulus for secretions of hormones and hypocalcemia inhibits secretions. It appears therefore that secretion is controlled by a feedback mechanism operating through the plasma calcium level in a manner similar to that of control of the secretion of parathyroid hormone. But in the reverse directions, the thyroid gland has certain interactions with the anterior pituitary gland. Thyrotropic hormones, thyroid stimulating hormone or TSH, that stimulate release of thyroxine. Thyroidectomy results in hypertrophy of the anterior lobe with degranulations of alpha cells and appearance of a beta cells, which morphologically exhibit certain alterations, so called thyrochromy cells. That secretion of thyrocalcitonin by parafollicular cells is dependent on blood calcium level and is not related to pituitary, thyroid and parathyroid functions. Now we'll see functions of parathyroid glands. The parathyroid produce parathyroid hormone PTH or parathormones. This hormone is essential in maintaining normal blood calcium level. The secretion of PTH is regulated by the serum calcium level. Through a simple feedback mechanisms, low level of serum calcium stimulates secretion of PTH. High level of serum calcium inhibits its secretion. Although the principal cells are believed to be the primary source of PTH, some evidences suggest that the oxophil cells may also produce these hormones.